Hey, good morning, everybody. It's my pleasure to introduce Boris Winterhoff and this lovely picture of a German Autobahn. Boris was born and raised in Germany, and he got his MD degree at the Christian Albrecht University of Kiel. Kiel is a really lovely city in northern Germany if you have the chance to visit. And he has a quite interesting CV because during this time he had these internships in several different US institutions, and among them he spent some time at the Whitehead Institute in Bob Weinberg's lab. He also went to Yale, and then he went to Mayo, and he said he fell in love with the programs here in the US, and he decided to do his residency and his fellowship at the Mayo Clinic. So Boris came to the U in 2014, and he is currently a tenure-track assistant professor in OBGYN and women's health, and his expertise is in single-cell analysis. He is trying to really understand the heterogeneity of ovarian cancers, and he has a very impressive CV. And he is currently funded by the Ovarian Cancer Research Fund Alliance and also by a University of Minnesota Grand Challenges grant. And thank you for being here today.
Thank you very much for the very kind and nice introduction. So good morning, everybody. So I'm a GYN oncologist by training. So I'm a cancer surgeon by training, but I happen to have also a strong interest in genomics. Had done quite a bit of work in the Cancer Genome Atlas project, and so some of the things that I kind of like to present here today, what we've been working on for several years during my time as a fellow at Mayo, and now we sort of transfer a lot of those things over here, is mainly in ovarian cancer and endometrial cancer. And this is, yeah, correct. This is a picture of sort of driving from, actually right here, you're still in the Netherlands, and then you sort of drive into Germany, and that's the German Autobahn. And sort of the title that I have here is On the Road to Precision Medicine and Gynecologic Cancers, ? And I like to take as a vehicle the German Autobahn, which we consider the mother of all roads, but that's debatable. All right, so I have my talk is structured into four subgroups, essentially. So we'll start with an overview of endometrial and ovarian cancer, and this might be repetitive for some in the room here. I don't know how much you guys know about ovarian and endometrial cancer, but I'll start with some clinical facts here and sort of the background and why we do what we do and what we try to accomplish. Then second part is lessons learned from breast cancer. So there's a lot of things that Dr. Yi and all the people that work in breast cancer have sort of achieved over the last decade and a half that we would like to reproduce in other cancers like ovarian cancer and endometrial cancer. And then in the third part, I'll talk a little bit about some of those things that we're doing right now with trying to sort of subclassify those cancers and try to stratify and come up with more stratified treatment approaches, sort of with the ultimate goal. We all want to do precision medicine and individualized medicine, but in my mind, clinically, we start out as one size fits all right now where we at. First step would probably be more like a stratified approach, kind of what was done in breast cancer. And then in the last section of the talk, I'll talk a little bit about where this can go and then also touch on single cell sequencing as, sort of one avenue that could help us to ultimately achieve that objective. So let's start with ovarian cancer. So ovarian cancer, compared to like breast cancer, for example, it's a fairly rare cancer. There's about close to 22,500 cases a year. Having said that, even though it's not that common of a cancer, is one of the deadliest gynecologic and female cancer. But as you can see here, of those women that will be diagnosed this year, 15,000 will ultimately die from the disease. So the 5-year survival rate very much depends on the stage, meaning how much the cancer has spread from the ovary to other organs, but it's anywhere between 30 to 92%. The problem with ovarian cancer, one of the big clinical dilemmas, is that there isn't any screening test or early detection tests. There also isn't a whole lot of symptoms that patients have early on. So the majority of those cancers we find in advanced stages, meaning stage 3 or 4, which is about 75% of those cancers. So over the last 3 to 4 decades, so what has been developed and pretty rigorously evaluated is treatment, and that's kind of what has emerged as a treatment paradigm that we use today, is a combination of chemotherapy and aggressive surgical debulking or cytoreduction. The biggest predictor for outcome is what my trade does, is do these big cytoreductive surgeries, which on average take anywhere from 6 to 12, 13 hours. And the biggest predictor is how much disease, residual disease, a patient has at the end of the surgery, which is, if you think about, I don't know any other solid tumor where that's actually the case. I don't know if Dr. Yi can think of any, but that's something that normally is not done for solid tumors, but for ovarian cancer, for whatever reason, we'll talk about those possible causes for that a little bit later. It really depends on how much disease is there at the end of the surgery. And you can see here those differences, 12 to 16 months overall survival versus 40 to 45. So this is still this is evolving right now. So now we're moving even to what's called R0 resection, meaning no visible disease. And those patients have in excess of over 10 years overall survival. So this is holding up even in 2017. So we classify ovarian cancer by histologic subtypes, meaning how those cancers look under the microscope. We have at least 5 different subtypes. It's the serous, endometrioid, clear cell mucinous, and the undifferentiated. Systemically, we treat them all the same. So there is a little bit of distinction. There is what's called IP chemotherapy, where you treat intraperitoneal versus intravenous. But the bottom line is where we treat them essentially all the same. So we do one size fits all. And that's not just true for gynecologic malignancies. That's true, unfortunately, for a lot of cancers. That you have one diagnosis and you treat them essentially all the same. So what are sort of the clinical dilemmas, and what are the things that we try to overcome with some of the work that we do and that other groups do? So despite all the progress in the biologic understanding that we have accomplished over the last decade or so, especially with the Cancer Genome project and other projects, still the majority of patients, more than half of patients, recur within 18 months and ultimately die within less than 5 years from this disease. So we haven't really made a whole lot of progress, if we're honest about it. So what are the things that we like to accomplish? Kind of what essentially most oncologists or clinicians try to accomplish. So to better biologically understand the heterogeneity of the tumors, and then try to find relevant pathways and try to sort of group those patients and find essentially the right patient, the right treatment for the right patient at the right time. So targeted therapy strategies, which sounds fairly straightforward, but it's clinically actually not that easy to do. So that's ovarian cancer, and those are the dilemmas with ovarian cancer. Let's talk about endometrial cancer. That's the other cancer that I spent most of my academic time on. So endometrial cancer is much more common than ovarian cancer. It's actually the most common gynecologic cancer with 61,380 cases. And interestingly, the incidence has gone up quite a bit. So when I first started doing this, when I started my internship, was about 11 years ago, the incident rate was close to 40,000, so it's about 20,000 cases more per year, which is pretty significant. The reason for that is mainly because of the rising obesity. So endometrial cancer, at least the lower grade, more indolent forms, are essentially a symptom of obesity. So they go up with if somebody has diabetes, if somebody, the higher the body mass index is, the higher essentially the estrogen levels peripherally through the aromatase in the fat tissue. So all this contributes to that. But interestingly, also the death rates have actually increased quite a bit too. So we're now up to almost 11,000 deaths. And if you go back 3 decades, the death rates were only 2,900. So even though the incidence has gone up, but the death rate has even gone up, higher than the actual incidence rate, which we don't have a very good explanation for that. Part of that is 3 decades ago, I think a lot of those cancers weren't accurately classified, so there might have been a mix with other cancers like cervical cancer, maybe even ovarian fallopian tube cancers. But still, that still doesn't really, explain that. I think what it says that we're probably not doing any better. I don't know that we're doing any worse than we did 3 decades ago, but I think we certainly haven't made a whole lot of progress. So clinically, luckily, the majority of endometrial cancers, like I said, are fairly indolent. So they're estrogen-driven tumors, and about 80% are stage 1, meaning they're limited to the uterus. However, even with those early-stage tumors, about 15% ultimately recur and die from the disease, which if you consider them clinically low risk, and we'll talk a little bit more about that, and that's some of the work that we do, you wouldn't expect, that high of a recurrence rate as well as death rate. So one of the things I think that indicates clinically that we still don't do a very good job in classifying those cancers based with standard pathologic criteria. And we can, I think there's an opportunity sort of similar to what was done in breast cancer to sort of classify them better. There's, for example, one class that we have something called high intermediate risk endometrial cancer, and that's based on the grade, depth of invasion of the tumors, lymphovascular space invasion. And I was about 2 weeks ago, I had a medical student in my clinic, and he said to me, he looked at me, he said, what does that mean, high intermediate risk? They're high risk or they're low risk? I said, yeah, that's exactly the problem. So we treat him as sort of high risk, but sometimes we do, sometimes we don't. So we'll give him adjuvant radiation, sometimes even chemotherapy. But again, if you look at that 15% rate of patients that ultimately die from that with early stage, supposedly low risk disease, I still think there's a lot of room for improvement. All right, so what do we know about the biology of endometrial cancer? So we molecularly classify them in type 1 and type 2 cancers. That goes back to the 80s, and that's mainly based on histology, but then also on some genomic alterations, like you can see here. So the type 2 are more like the high-risk ovarian cancers. They have p53 mutations. They're normally high-grade serous. Those are just the histologies. And the type 1, the endometrioid, the more indolent, hormone-driven tumors. So you can see where this is going. So we also classify them histologically, but we do the same thing. So in GYN oncology, we have a tendency to treat everybody with carboplatin paclitaxel, which is not that we haven't tried a lot of different chemotherapies that happen to be, the most effective treatment that we have. But it's sort of the same. It's sort of the same approach. So we have the one-size-fits-all approach. And again, sort of the same things that we've talked about in ovarian cancer. So we have learned quite a bit more about the biology. There is now sort of approaches to molecular classify those cancers and then trying to find the right group and give the right treatment rather than this one-size-fits-all approach. So it's coming to the next step. So we have taken the Autobahn sort of from the Netherlands. So now we've gone over all the way to the far northeastern end of Germany. So this is an island called Usedom, which is in the Baltic Sea. Over here is actually Poland. So this is right at the Polish border. It's a very, very beautiful place. If any of you ever get the chance to get there, they have maybe the most beautiful architecture anywhere in Germany, right on the coast. The German emperor used to go with his friends back in the 1800s, and they have built these massive places right on the shoreline. It's really pretty. So what can we learn? What can we learn from breast cancer? And again, so one of the things we try to do, things have worked out, very well in breast cancer, using some of those approaches. And that, again, is one of the things we're trying to do to essentially replicate that in ovarian as well as endometrial cancer. So they have achieved molecular subclassification using cDNA microarrays, and that was back in the early 2000s that they've started with that. That was used not only to molecular subclassify patients and come up with different treatments for those subgroups, but also as a prognostic tool. And those are things that we think can be directly translated to ovarian as well as endometrial cancer. And then also they did quite a bit of work on identifying signaling pathways and specific targeted treatments. So this brings me just back to so really everything sort of starts with the basics, DNA, RNA, and protein. And this all really goes back to Watson and Crick back, it was, I think, 53, and then they got the Nobel Prize in 62 for discovering of DNA. But that's really what is the basis for these arrays that people have used. And that's already outdated. Now everybody uses RNA-seq, but the principle is the same, that you look at gene expression, you do that over all 20+ thousand genes, and use that to classify and come up with different molecular groups. I can jump over that because nobody's doing microarrays anymore. So molecular subclassification. So the first, like I said, so some of the first papers that came out were in the early 2000s and did essentially an unsupervised clustering approach. So you take 20,000 genes, and then you essentially just have the computer try to come up with groups that are similar biologically. And if you talk to, I don't know how many people are bioinformaticians in the room here, but if you talk to somebody today, they tell you, well, that's not how you would do it. But interestingly, because we have started with this one-size-fits-all, this is for sure not the best approach to analyze these data, but clinically these things still hold up. So, what does it tell us? So maybe that approach is better than we think. I think what it tells us is that bar is probably so low because we grouped them all together or overtreated or undertreated patients, that these things hold up and are used now clinically. So there's these different subtypes, Illumina A, B, HER2 positive, basal subtypes, that they have different treatments. The thing that I think is even more interesting, and that's especially for endometrial cancer, by the way, we sort of moved now, so we're from the northeastern part of Germany. Anybody knows where that is? Sort of in the south of Germany. So that's Marienplatz in Munich. So those are some of the Hofbräuhaus umbrellas here. So the prognostic potential of using these arrays, I think, again, is for endometrial cancer would be quite interesting and attractive to try to replicate. So what have they done for breast cancer? So they started, that's Laura van der Vaart's group and René Bernhardt's out of Amsterdam. So they started to take early-stage breast cancers, and they looked and tried to come up with cases that have higher risk of recurrence versus some that have lower risk. And most of them were node negative. Some of those were node positive. But that's a similar sort of scenario that we have. If you remember one of the earlier slides, I said majority of endometrial cancers are early, but there's 15% of those early stage low risk that ultimately recur and die. So what we do right now, the options that we have without having a classification like that, so either you don't treat any of those, knowing 15% will recur, you could treat all of those, meaning you would overtreat 85%. And that's sort of where they were at breast cancer. So they probably, and Dr. you correct me if that's not correct, but, before these classifications were there, majority of them were either over or undertreated, depending, I guess, how you look at it. So you'd either radiate everybody, same as kind of with taking out lymph nodes. We have sort of the same dilemmas, essentially, in endometrial cancer. So they then went once they had identified their subgroups and their genes, they reduced it down to 70 genes, and then they validated in 295 cases. And then they compared it to the gold standard, which in Europe are the St. Gallen criteria. That's a place in Switzerland where they meet, I think, regularly every couple of years to come up with pathologic criteria to classify or risk stratify breast cancer. There's NIH criteria, and then here you have the 70 gene set. And they had this famous paper in the New England Journal where they compared those and showed that their classifier seems to outperform, at least in this initial data set, the pathologic criteria from either St. Gallen or NIH. And that's the test here. So the MAMA print is probably more commonly used, I think, in Europe. That's again, that based on 70 gene panel. They did get FDA approval. There is this test here in the US called Oncotype DX. Dr. Yi, have they ever gotten the FDA approval Oncotype DX?
Yeah.
No. I think they did, right? And they did, right?
Yes, they only do it in California.
Okay. But still, most people use it, right? Yeah. So that's, I think, one thing that's very attractive, and that's essentially exactly what we're trying to do right now for endometrial cancer. Let's go to the third part, I think, that we could really learn from breast cancer. And sort of we've sort of moved away now from Munich, sort of further south. Anybody an idea where this is? So that's sort of the Autobahn driving into Austria. So this is Austria here, and those are the Alps, and that's going south towards Italy. So the mother of all targeted treatments is probably HER2, and that's sort of, you can argue about that too, but that's probably the biggest success story, I think, at least in solid tumors. So that was back in 1987. Dr. Slayman and colleagues published that, and I think Dr. Yi was quite involved with the whole HER2 story as well. So they started out and found HER2 gene amplification. They saw then that translated to overexpression on the protein level, and that also translated into prognostic relevance, so those patients actually had worse outcome. So about 14 years later, they then did a clinical study where they had a monoclonal antibody against HER2, Herceptin, and showed that those patients and if I recall right, that I think wasn't that study initially negative, and I think they went back and just stratified based on HER2 expression, or the subset, right? And which that's really sort of the proof of concept here, that if you do these studies, and that we have the same dilemma in sorry that you had to get a mic now, keep asking you, but so we have the same dilemma, and I'll show you that in a little bit in ovarian cancer. And pretty much, I would actually claim in most tumors where you use a targeted specific treatment, if you do unselected treatment without enriching for a subgroup, they most likely will be negative studies, depending on what your readout is. But if you want to have 20% or more response rate, I think there's a high chance you'll have negative studies. And there's plenty of examples where people have done large phase 3 studies for millions of dollars and have essentially negative results. The problem is, clinically for us, a lot of times we don't know how to enrich. So sometimes we have these really good ideas and think that we could enrich, a certain way, and then that doesn't hold up. So there's essentially clinically, I think, only two strategies. So you either decide before and have a really good rationale why you enrich for a certain subgroup, or you do the studies this way, you treat everybody, and then, sort of retrospectively try to come up with the right subgroup, which both approaches are not ideal. But, we hope that with more understanding biologically, we could get more towards, and the match study is sort of a good example. We'll see whether, that ends up, being positive. But, that's sort of, one of the approaches that's tested right now to enrich essentially up front rather than retrospectively. So that brings us a little bit farther. So now we've sort of driven through Austria, and that's a large lake in northern Italy called Garda Lake. So that's a city called Torbole. That's sort of the northern end. If anybody likes to windsurf or kite surf, so that's like one of the best probably lakes for that, because they have these large. It's quite spectacular, actually. They go up here almost 5,000 feet straight up these cliffs here, and they have quite a bit of wind there on a daily basis. So what have we done in ovarian cancer? So it sort of all started with a lot of the capabilities that were done in the Human Genome Project, and that's now 2001. I still remember when that came out. I was just barely in medical school, but yeah, that for sure, I think, had a large impact on all of us. And it's now 16 years ago. So that's one of the things this led to was the Cancer Genome Atlas project, which was a large NCI-NIH project that tried to bring some of these advances that were made with the Human Genome Project to larger scale. Diseases and cancer was one of the first ones. So the idea here was, and that's now accomplished for a lot of different cancers. I was involved with the ovarian part of it, and the endometrial cancer and carcinosarcoma part of it. But the concept here essentially was to take some high quality frozen tissue, take blood from about 500 plus patients, have matched pairs, and then essentially do any sort of genomic analysis that you could do at the time. And again, so that has sort of evolved throughout the project, which again is now completed. And now we're sort of we have now sort of there's a project we're doing right now. It's called Pan-GYN, where we now, after all those projects are done, we're trying to take now all GYN cancers together and compare them to other cancers, breast cancer, for example, to try to come up with hypotheses and possible clinical trials. So one of the things that for ovarian cancer came out of the Cancer Genome Atlas project was similar to what I've showed you earlier in breast cancer. So about 10 years later, we had a first sort of attempt to have a molecular subclassification of ovarian cancer. Similar approach. And those were done also still with arrays initially. So you take all 20,000+ genes, and then do essentially unsupervised clustering, have the computer come up with subgroups that have similar biology, which led to 4 subgroups. And interestingly, and I had that explained to me a couple times, but I don't think I can explain it very well. But most of the times, by chance, you come up with 4 subtypes. They've looked at 2, 3, 4, but 5, 6, and more. But that, for whatever reason, a lot of times there's some bioinformatics reason for that. So that's what we had here too. And then those subtypes were essentially named based on the majority of genes that were overexpressed in there. So you have a differentiated subtype, then you have an immunoreactive. That's called that because there's a lot of infiltrating immune cells, and that's why a lot of those genes are upregulated. And you have a mesenchymal and proliferative subtype. So that was published in 2011. The problem was that the clinical follow-up is pretty poor in the TCGA data sets, and a lot of you guys probably know that if you work with these data sets, and that's just because most of those samples were collected prospectively. There isn't very long follow-up, and by the time the project and the accruement completed in 2014, most people don't update those clinical follow-ups, which is kind of a shame, but that's why those follow-ups weren't very good. So when they looked whether there was any prognostic relevance of those subtypes, they couldn't show any because just the follow-up wasn't long enough. So that's where we came in. So we then took a cohort from the Mayo Clinic that had 10-plus years follow-up, fresh frozen tissue, 173 cases, and essentially just reproduced it. So we took those, they used 1,500 genes to come up with those 4 subtypes, so we just took those 1,500 genes and just reproduced those 4 subtypes. And interestingly, and this is overall survival, so we could show that there's significant difference in overall survival, so kind of did the first step and said, well, is it prognostically relevant? And if you look at this here, that's the mesenchymal subtype and the proliferative subtype. So those do significantly worse compared to the immunoreactive does the best, which makes sense. There's some other work supporting that. If you have infiltrating immune cells, you probably have a more active immune system there fighting the cancer, and that might be the reason, it's probably the reason why those do better. The differentiated are normally lower grade tumors. That also makes sense. So that's ovarian cancer. And then how about endometrial cancer? So I told you earlier that one of the big problems is that even though majority are clinically low-risk patients, there's still 15% or so of patients that ultimately recur and die, even though they're clinically low risk. So they've done the same, well, I guess it doesn't hold up here. So here they have 3 subtypes, but so that's what we did with TCGA, same thing. And here we could actually show at least progression-free survival difference. We're actually right now repeating that with 168 cases from the university here with also 10-year follow-up. So same what we've done for, I don't have the results yet, but I'm sure that there will be also a difference. So it brings me to one other marker I want to present here. So this is some work I've done together with a group from Germany, Austria, and Israel. So they have identified a marker that they originally described in pancreatic cancer. I've been working on that for 25 years now. It's called L1-Chem. What that is, it's essentially an adhesion molecule that's mainly described in the brain. But they have tested that across a lot of different solid tumors, and one of them, of those tumors that they tested, was endometrial cancer as well as ovarian cancer. And whenever those cancers were positive, you could see, much worse clinical outcome and survival. So we've then just taken a large cohort of endometrial cancer, stage 1, because again, that clinically is sort of the big problem to identify those 15% or so. And we stained them for L1-Chem. And you can see this is univariate analysis. So those are pretty impressive curves. I don't know how often you guys have seen curves like that, but that's pretty impressive difference, how you can differentiate just between having positive stain versus negative stain. This is now going to overall survival, still univariate. And what's interesting here, so the things that we use right now to come up with this high intermediate or high risk cancers is depth of invasion and grade of the tumor. And somebody that has a deeper invading tumor and higher grade has worse outcome. But if you then factor in the L1-Chem, so even tumors that have deep invasion but are L1-Chem negative, have over 90% of them are still alive after 10 years. So that's sort of essentially against sort of what we right now classify them. But if you then at the same time take these high and low risk classifications, so take all these factors together, depth of invasion and grade, you can also see that the L1 chem outperforms that. If they're negative, they will be alive. If they're positive, even if they have good features pathologically, they most likely will not survive 10 years. And if you then do a multivariate analysis, so you have hazard ratios of 15 as well as 13 for overall survival, while things like grade, even high grade, is only 2. So that, again, this is 1,000 cases. This has been externally validated now. We've actually validated that now in 168 cases here. A friend of mine in Germany has validated in over 500 cases. So this holds up. The problem with this is clinically, and I don't know if any prognostic immunohistochemistry marker that's used clinically. I know, Dr. Yi, if you know any, but I don't know if... Yeah, well, yeah, you could, right. But besides that, it's technically pretty difficult, and it took us about 1.5 years to establish that stain here. And the reason is, so the definition of positive is more than 10% of any stain, but that gets pretty subjective. If you have different pathologists, and one says, well, that's 9% or 8% or 12%, we've tried automated systems. It's not that easy to reproduce. And for a clinical test, you got to be pretty accurate, because if you make treatment decisions based on that. So even though this, I think, is a phenomenal marker, it'll be difficult to establish that clinically. And that's kind of where some of the work comes in that we're doing now here. So we've sort of looked at the TCGA, and even in the TCGA with the poor follow-up, if you have altered L1 chem, you have worse outcomes. So that even holds up there. The other interesting thing here is, even though we know it's prognostically relevant, we still don't know whether it's really a driver and really something that drives the disease. But if you do network analysis, and I just did that last night, so excuse the sort of convoluted slide here, but it is linked to quite a bit of different pathways that we actually are actively pursuing now with clinical trials. So FGF is in here, EGFR is in here, PI3 kinase. Those are all major pathways in endometrial cancer. So it is certainly possible that L1 chem is responsible for the worst outcome, but it might just be sort of a passenger sort of marker that, for whatever reason, it's actually a marker that's associated with high-grade serous histology, which is one of the worst sort of histologies. So it could also be that this just identifies subclones of high-grade serous and otherwise low-risk endometrial cancers, and that they're missed on regular H&E, which if it's 10%, that's certainly possible. And that could be another reason. So we don't really know, again, whether that is really a driver and whether that's something that could be drugged. The German group actually has a monoclonal antibody in phase 1 right now against L1 chem. But again, we don't know whether that's going to hold up or not. So and again, that's kind of where we come in. So we have 168 cases here that we have done adjuvant gene expression array, fresh frozen tissue from the University of Minnesota. And so what we're doing right now, we're trying to essentially do the same thing they did for breast cancer with those 70 genes, or the Oncotype DX with 21 genes. We'll take the patients that are L1 chem overexpressed on IHC in that cohort, the ones that have positive lymph nodes, and the ones that recurred, and sort of classify those against the ones that don't have that, with the idea to come up with essentially a list of genes, a gene expression classifier that we could use then on a nanostring platform to identify the ones that are really high risk, those 15% that, in those stage 1. And of note, the L1 chem actually was 17% out of those cases in the stage 1 that were positive. And when I told you earlier, it's about 15% of the stage 1. So they might find the high-risk group. But again, because of technical difficulties with reproducing, I think it would be much cleaner to have sort of a nanostring-based gene expression classifier that you could run on paraffin, hopefully much more reproducibly than the IHC stain. All right, that brings me sort of to the last part of the talk. So we've now gone from that Garda Lake in northern Italy a little bit farther south. Anybody knows where this is? That's in Venice, so we're kind of moving further south in Italy. So how can we now, so I showed you earlier, we're switching now back to ovarian cancer, so how can we use those molecular subtypes? So we have shown that they have prognostic relevance. But now the next question is, even though that's interesting, clinically, to be honest, in ovarian cancer, I told you earlier, the majority of patients recur and die from the disease, even if you can find a subgroup that is has a little worse outcome than the other. clinically that doesn't mean anything because all of them get treated with the most aggressive treatment that we have. We wouldn't withhold any treatment at this point for somebody who has stage 3 or 4 ovarian cancer. So then the next question that we asked, we said, well, does it have any predictive relevance? So can we identify subgroups that respond different to specific targeted treatments? And we were fortunate that we were able to get some samples from some collaborators of mine in Germany as well as the MRC in the UK that have conducted a large trial in ovarian cancer. It's called ICON-7. It's a phase 3 study, 1,500 patients, where they had one group that was treated with standard carboplatin paclitaxel, and the other group was treated with carboplatin paclitaxel and Avastin, which is a monoclonal antibody against VEGF-A. So they collected I got 1/3 of those samples from the Germans, and then they shipped them over to Rochester because that's where I was at the time. And then we isolated RNA out of those samples and DNA and microRNA, and then sent that to San Diego to Illumina, and they did DASL gene expression arrays as well as methylation arrays in those patients. So this is this is the design of the study. So again, 1,500 patients, frontline phase 3 study, carboplatin paclitaxel, and then plus Avastin in the experimental arm. So this was the result of the study, the progression-free as well overall survival. And I don't know how often you see these Kaplan-Meier curves, but what you want is that they really separate. So those don't separate at all. There's 2.4 month progression-free survival difference. And technically, it's a positive study because the primary endpoint was progression-free survival. But if you ask any clinician, 2.4 months progression-free survival difference for a treatment that costs about $120,000 a year, and 3% of those patients get bowel perforations, that's a pretty difficult sell, I think, for a patient to get a treatment like that. Overall survival, same thing. There's essentially no difference. Interestingly, and I'm not going to go too much into that because it gets pretty controversial, but they did a post hoc analysis that they didn't plan. They tried to identify the right group, so a high-risk group where they said, can we identify a group that really benefits from Avastin? And what they looked at clinically high risk, which those are the patients. Remember, one of the earlier slides I showed you, the biggest predictor for outcome is how much cancer is there at the end of the procedure. So the patients that had more than 1 centimeter residual cancer, which that's called suboptimal, as well as all stage 4 patients. So those are the most advanced and worst outcome patients. When they just looked at that subgroup, they saw a 5.5 months progression-free survival difference, and they saw a 7.8 month overall survival difference. So now we're getting to something, that's clinically relevant. And you can argue about is 6 months or 4 months or what is relevant. 7.8 months, I would say most clinicians would say that's a significant difference. So based on that, they actually got approval frontline in Europe for that. So it's used frontline in ovarian cancer in Europe in this high group, the suboptimal stage 3 and stage 4 patients. And the controversy comes in, and I don't want to go too much into that. There was a similar study done in the US called GOG218, which essentially was enriched for this high-risk group, and they did not see the difference. The difference between those studies was this wasn't placebo-controlled. The US study was placebo-controlled frontline. Otherwise, exact same studies. I have a hard time believing that there's placebo gives you a 7.8 months overall survival difference, but they still didn't get approval for that here. So the FDA said, no, the US study is negative for that. We're not going to approve that here. And that's all I'm going to say about that. Those are 400 patients. I think close to 400, 380 or so. Yeah, that was, there were much more in this. So the US study wasn't 1,500, it was 1,800 patients. And majority of them, that study was just open for that subgroup, the high-risk clinical subgroup. So it was much higher. I think it's over 1,000 in that group that met those criteria. The other difference, they've used twice the dose of Avastin. But if you use more in the US study, if you use more, they had the PFS overall was 3.8 months. So they got about double those numbers. Yeah, it's nobody has a good explanation for that.
Did they go back and look at things like BMI or anything like that?
No, and it's not very relevant in ovarian cancer as far as we know, because most of them aren't really hormonally sensitive. If that's kind of what you're thinking. Yeah,
related to obesity, right?
Sorry.
Yeah, but it's no, not that I know of, but most patients aren't obese with ovarian cancer. Pretty rarely are they obese, and yeah, we don't really know that that's a major driver.
So, when this debate has come up in breast cancer between the U.S. and Europe, there's always been the quality of staging as a suggesting the difference. So the argument is, I'm not casting aspersions on our European colleagues, but the argument has been
fair.
European
we say that
surgeons
all the time.
were understaging their patients. So there was a stage migration phenomenon that accounted for the difference rather than true value.
Yeah, we don't know that actually the optimal, the bulking rates are higher in the European studies. So if anything, they maybe got more aggressive surgery, even though we always say we know how to operate better, but you know how that is. I don't know whether that factors in, and it gets even more complicated. I don't want to get even too much into that. So I'll show you in a minute here, and I took those slides out because I don't think it's necessarily relevant, but we're talking about it, so let's talk about it anyways. When we looked in that German subgroup, those 420 patients that we had, we tried to reproduce this. And so if we started with all of those groups, there was actually a 6.5 months progression-free survival difference in the unselected German group. And then when we looked at that high-risk group they have here, there was no difference. It was 6.6 months. And that's just for PFS. I didn't have the mature overall survival difference. So one thing, it seems like that for whatever reason, Avastin works much better in the German patients, which nobody has an explanation for. It gets even worse. So the rest of the study, the remaining 1,000 patients, is essentially a negative study besides the German patients. So for whatever reason, it's all driven by the German group, and that is sort of known but not published. I don't have an explanation for that. I don't know if the pharmacogenomics is different in German, and for whatever reason they respond better. There were 93 different centers in Germany that participated in that study, so I have a hard time believing that they, for some reason, stored it different or there was some other difference. But yeah, it's a little troublesome, but...
So, in these studies, being totally naive about this, how often do they do pharmacokinetics, if at all? Because this brings up a really big point. I mean, that I think even colleagues that I know that have started to do pharmacokinetics, you're always going to have this kind of question, right? Mm-hmm. And so, as a general rule, is that just not ever looked at?
Yeah, that is an excellent question. I might have to refer to Dr. Yi again here. But so interestingly, if we just talk about this study, so this was a large study that was, I think, close to $30 million sort of like budget for that study. There wasn't any systematic, really, biomarker or pharmacogenomics built in. So we got lucky to get those 400 some, essentially all the German samples, because they had a sub-protocol in Germany where they actually wanted to look at histologic accuracy of the pathology, and they did an external review, and that's the only reason why we got those paraffin blocks. But yeah, no, they haven't in those studies. I mean, I think now going forward, more and more studies probably systematically try to at least collect blood and paraffin tissue. But logistically, if you talk about 93 different centers, that means they're not all academic centers. So we've done some studies with a network where we only included centers that can provide fresh frozen tissue. But even that is not that easy, to provide that on a high quality level, meaning 20, 30, minutes sort of snap freezing after it comes, into the frozen section lab. Those things aren't that easy to do. But no, I agree with you 100%. I mean, if you have a budget of that, it's always strange to me why it's not factored in unless you don't want to do that for some reason. But I don't know your thoughts on Dr.
I divert too far, but it's something you argued a little bit earlier, that in many instances they do not want to develop a companion but diagnostic to the drug. That gets to be very expensive and difficult regulatory pathway. So, if you had a marker like L1-CAM, for example, that was a predictor, and it was hard to actually do the assay, then you would rather have a drug you can give to everybody, and it would be a positive result.
Sure.
So I think that actually that's one of the things that companies struggle with, is that, and particularly there were no preclinical data that were a predictive biomarker for Avastin. Right. So they really
I mean, until this point, I mean, this I mean, we just published this, but this is not validated. I mean, we're working now to get in other studies to validate this. But yeah, right. So I mean, people have tried pretty hard, and if you read the literature on like colon cancer and stuff, I mean, there isn't hardly anything they haven't tried. Pharmacogenomics, actually, I think they have done some in that GOG 218. They had some blood samples, but none of that, I think, held up.
And probably part of it is that deciding what you would look at. Mm-hmm. So if you wanted to do pharmacokinetics on, how effective Avastin was in two different populations, what would you be looking at the board? I mean, you'd have to know enough about that drug and understand its metabolites and have that chemical knowledge in order to really know whether there was a group that was more responsive. Or even to look at like VEGF mutations or VEGFR mutations that might, downstream cause a difference in response to some of these drugs. So it probably does get way too complicated to even start going that route.
So we coming back to those German samples. So we essentially did the same thing we've done before with the Mayo cohort. So we just reproduced those 4 subtypes, and the idea was because we've showed in that cohort from Mayo that they're prognostically relevant. The other thing we also looked at is what are the genes that define those subgroups? Are there any that have sort of pro-angiogenic gene lists? And it happened to be that those 2, the proliferative and mesenchymal, are the ones that have things like VEGF, angiopoietin, and things like that upregulated. So our hypothesis was maybe those are the ones that derive more benefit from treatment with Avastin compared to the other ones. And that was those subtypes when we did those. That in itself is pretty interesting because nobody has ever done that in a phase 3 study. But they turn out to be about 1/4, so they're pretty evenly distributed. And the one thing that we first looked at was to see whether, because I showed you earlier, there's this high-risk group that they've identified in this study. Are these molecular subtypes just a fancy way to find those clinical high-risk group? If they are, well, then it's great to do, but I don't know that there's a whole lot of clinical value in that. But interestingly, it isn't. So you identify a significant amount of other patients, if we go by this hypothesis that proliferative and mesenchymal are the high-risk group, and those are the ones that have the more angiogenic sort of signatures. And you do identify another almost 60% or so that you wouldn't have identified with that. So that, we were happy when we saw that. And then we just applied those molecular subtypes to the progression-free survival. And the overall survival we're just about to get, and it just is mature, so we haven't really worked with that. So that maybe discard that for now. But so for progression-free survival, the proliferative subtype had 10.1 months progression-free survival difference. And that clinically, again, this is not validated. That's a really small subgroup. That's a retrospective analysis. But at least as hypothesis generating, that clinically is a range once you get over 6 months. There's a lot of sort of cost-effective analysis with Avastin ovarian cancer. Once you get over 6 months progression-free survival difference, that might be clinically relevant. Then we looked at the mesenchymal subtype, 8.2 months, not significant. They end up crossing those curves, but nevertheless, there might be a difference too. And interestingly, because you almost have to expect that if those other ones have more benefit, while the other ones almost have to have less, and they sure enough do. And that clinically also is interesting that there is subtypes that might benefit less from it, and those are the differentiated and the immunoreactive subtype. So that's just sort of the summary of this. So maybe the proliferative and maybe the mesenchymal subtype are the ones that might derive more benefit from it. So those are essentially just the conclusions. So if you try to now sort of take that one step further. So the first thing, and we're working on that right now, so we hopefully can get 400 cases from a different study that was done in the US to validate that. There's a GOG study called GOG 213 that I have verbal commitment to get those samples. We need to get that in writing, but hopefully we can get those and then at least start validating that. But if you want to take that even one step further, so you could envision something like this. You could take patients with ovarian cancer and then operate on them, take the tumor, and then classify them in one of those 4 subtypes. And the mesenchymal, as well as proliferative, like I said earlier, could have angiogenesis treatment. Differentiated, we don't really have anything that we can come up with, but maybe we can come up with something standard chemo. It could be a control arm. And then the immunoreactive subtype, again, angiogenesis inhibition doesn't really seem to help, but now there's a lot of hype about immunotherapies and PD-1 and PD-L1 inhibition that has been tried in ovarian cancer like it has in a lot of other cancers. The overall response rate is anywhere between 11 and 12%, which isn't enough that any company is interested to take that forward. Unless you get to like a 20% overall response rate, you most likely will have a negative study if you do unselected patients. So we were actually able to, and I have that a little bit later, to work with Merck on sort of enriched approach here with PD-1. So that brings me to one of the projects that we have ongoing now here. So we got some funding from the Grand Challenges program at the University of Minnesota. And so what we're trying to do with our project is to now try to put some of these tests actually into clinical practice. So to start a precision medicine program in ovarian cancer. And we're trying to start with 30 patients. And we actually already have the first patient enrolled. We'll have another 3 this week. And what we're trying to do is to essentially take blood from all those patients, take fresh frozen tissue, take paraffin tissue, then do exome sequencing of their germline, do exome sequencing of their tumor, do RNA sequencing of their tumor, then also do nanostring to sort of kind of refine that classifier for those 4 subtypes that we're working on. And then trying to integrate that with, because that's, I think, really where the rubber meets the road, trying to integrate that into sort of clinical data. So to take the electronic medical record, build a database, and automatically have genomic data and automatically pull data from Epic. So that's, unless we'll build some infrastructure to do this automatically and systematically in patients, it'll be very difficult to envision how you're ever going to use any of this clinically. Because somebody like me that sits in clinic, I mean, I happen to have an interest in these things, but a lot of people might not, and it needs to be, you really have to have one page, in your electronic medical record where it says, this is the subtype, this is how you treat it. You have to, unless we sort of build and start with that infrastructure, and that's just what we're trying to do with those 30 cases. And that's just sort of, just a fancy chart of that, but that's essentially what we're trying to do, and then really trying to integrate that. And we sort of that's still work in progress, but there is this C-Bio portal, which was done by a friend of mine, Nikki Schultz at Memorial Sloan Kettering. He's coming actually in June to speak to the precision medicine group, and that could be one avenue that we could pursue to have something like the C-Bio portal that has done, I think, a nice job to sort of make TCGA data clinically usable and essentially have like a one-page summary. This is altered in this tumor. Those are the potential drug targets linked to clinical trial databases. Whether it's going to be that or something else, I think that's sort of really what clinically for us would be the most useful if we could have something like that. And then the other thing that we're right now, we're working on opening a study that Mayo has through their SPORE project frontline study. So the other thing we want to do, all those patients that are on the precision medicine program, those 30 patients, ideally also go on clinical trials because we will get much better sort of clinical follow-up data through that. So that's sort of the other thing we're working on. And again, that really comes back to, as a starting point, trying to come up with ways to use those 4 molecular subtypes. And again, 10 years from now, that might be, we might look at like we look now at the one-size-fits-all. But you have to start somewhere, to try to do these things. The other thing that's exciting, so we'll work with Gottfried Konechny at UCLA and then Andrea Warner-Hendrickson from Mayo. So we will be opening a study with Merck where we actually try to use those subtypes and enrich for the immunoreactive subtype and treat them with PD-1 inhibitors in recurrent ovarian cancer. With that idea, we don't know whether that's the right subtype, but we know if 11% response rate, nobody is going to take that anywhere. Maybe if you take the ones that already have all the infiltrating immune cells, those are the ones that are, we don't know whether that holds up, but that's one, I think, idea that could be pursued, and we'll be doing that. So the goal is here we have enough money to do 30 patients, so screen 120, and then take 30 patients and then treat them with the PD-1 inhibitor. And that brings me sort of to the last sort of part of this presentation. So everything I've told you so far is using bulk tissue. which I think for certain things is good, and that's sort of all we had at the time. So if you think about those molecular subtypes, I think it's actually helpful to get a general gestalt and try to find different subgroups, because you need the gene expression probably of the stroma cells, probably of the immune cells, to come up with these sort of broad different groups. But if I told you earlier that for ovarian cancer, the main problem is that the majority of patients ultimately recur from the disease. By the time they recur, there's no cure that we have. And not people haven't tried any chemotherapy you can imagine. So that leads us to believe that there is two options. So one, there could be some cells there that we either don't get out mechanically with surgery and that are resistant to chemotherapy from the beginning, which some people call them cancer stem cells, like Bob Weinberg does, or like multi-drug resistant cells, or whatever you call them. We can't eradicate those cells with the treatments that we have. So either those are there from the beginning, which we believe is the case. The other option is they could develop throughout treatment and ultimately develop and are not there from the beginning. The first scenario would be the better scenario, I think, because then we could identify them and hopefully come up with some way to treat those. If they develop after treatment, well, that might get much more complicated, but that's still, would be sort of the second scenario. So, and that's a lot of work that I'm doing with Tim Starr. So what we're trying to do to go from this bulk sequencing to sort of the single cell level with that idea, if let's say really that's true that those cells are there from the beginning, they're probably pretty low in numbers. So they're anywhere maybe 1 to 3% in the original tumor. That's at least some of the initial work that has been done with those cancer stem cells. So if you take a bulk piece of tissue and sequence thousands of cells, you're not going to find a signal of 1% of the population. That's just statistically impossible. So the only way to overcome that would be to have technology that can go down and do the same analysis on the single cell level. And Dr. Sachs has done a lot of that work in liquid tumors. So, we sort of piggybacking a little bit on some of the advances that have been made there. But we have been able to successfully, we just published the first patient we've done in ovarian cancer where we did this single cell sequencing. And that was initially only we had essentially 66 cells. But one of the first things that we already learned, we did those subtypes again. And the one thing we learned, and you probably would have expected that, well, one patient actually has more than one subtype. So if you actually go down to a single cell level, well, that's actually not that simple anymore to classify them. Clinically, it might still hold up, but again, that's because if you start out from one size fits all, anything that's better might give you some clinical benefit. But that's sort of one of the first things we learned. The other thing that was interesting, and then again, this is 66 cells, and this is only one patient. So I don't know if we can sort of draw too many conclusions from that. But there is a bunch of stem cell markers published in ovarian cancer, and there was one cell that indeed had all of those markers upregulated. Whether that's a stem cell or not, we don't know. We parallel have avatar models in those patients. Unfortunately, that patient, the tumor didn't take. So the idea was, and that we got some funding now from the OCRF to do 30 patients, where we have avatars at the same time, treat them, make them, platinum or make them resistant, essentially, and then do single cell sequencing again, and then hopefully have a pure stem cell population, and then we can go back to that original patient. And we just finished the first patient, and we'll actually have, like I said, another 3 patients for the precision medicine, but they all happen to be in the single cell study too. So they're like in 8 different studies now or so, those patients. But so what we hope that now, because we're using a different machine, so with that last patient that we did, we had 96 cells, so we'll do exome sequencing on a single cell level. And then they have this new machine called 10X. We don't know yet how many cells we'll get, but it's anywhere between 3,000 to 10,000 that we can do RNA-seq on. So now we'll get into a number that might actually be able to find a 1 to 3% subpopulation. I mean, with the 66, well, that's sort of chance. But if you now talk about, even if we only get 3,000, so that might be enough to identify that. We'll see whether that holds up. The other thing we'll do with that, too, is if patients actually recur, then we'll biopsy them again and do single-cell sequencing again. So then we have sort of the avatar avenue, but then hopefully the patient doesn't recur. But if they do, we would also have that to go back and see whether we're right and those cells are there from the beginning. again, the second possibility is they are not there and they just develop, which that makes things even more complicated. But let's hope it's the first, because I think that'd be easier to target. And that's sort of the concept that we're trying to do here. So we still need the surgery to remove the majority of the cells. We still need the standard chemotherapy, which, again, hopefully can eradicate 99% of the remaining cells. But if we're right and those stem cells are there from the beginning and that's why they recur, well, then we need some anti-stem cell agent, and whether that's sort of like, I mean, there's things like aurora kinase inhibitors. There's a couple of possible targets. The next sort of difficulty will be, even if we can identify those cells, so hopefully they have all the same sort of biologic pathways. We don't know that yet either. I mean, it could be they all have different sort of pathways. Well, that makes it even more complicated. But unless I think you can actually identify them on that level, it'll be very difficult to, with bulk, sequencing, I think it'd be impossible to, make any progress, at least if that theory is right. And that's sort of the summary. So really, we have learned a lot genomically, but I think sometimes people forget that what we do clinically is still a lot of those things. I mean, it's all sounds good in theory, but it's much harder to actually translate that. And I think one of the first ways is to do sort of a stratified approach and just have broad sort of bins and molecular subgroups for targeted treatment. And then, ultimately, I mean, that single cell that if that pans out, that would be great. But that's still a little bit down the line. And those are just, again, sort of targeted therapies. But then at the same time, for example, for the endometrial cancer, I think also very relevant to find the groups whom to treat and whom not to treat. So I said earlier in breast cancer, they used to overtreat a lot of patients. I think now they have those classifiers and they do much better. For endometrial cancer, so one thing I didn't tell you, sort of the U.S. approaches will do lymphadenectomies, mainly taking out lymph nodes in pretty much all endometrial cancers. They're only positive 12% of the time. So that means we're taking them out in 88% of patients that wouldn't ever need that. So that's one of the things that we look at with that classifier, positive nodes, is sort of a bad predictor. If you could just half that number, because not only is that expensive to do, but there's also complications like patients get lymphedema about 25%, which that's pretty miserable. So if you can avoid that and sort of optimize that would be great for patients. And the Europeans have a different approach. They don't take out a lot of lymph nodes. They just radiate everybody. So they radiate pretty much all of their early stage endometrial cancers. But if only 12% have positive nodes and only 15% recur, that also means you overtreat 85%. And radiation is not only expensive, but that also can cause secondary malignancies down the line and a lot of complications, bowel complications, things like that. So that's the team, and this is all this is a large team that we have together here, and that keeps growing and growing, which is a lot of fun. It brings me to the last slide. So we went from Florence. You guys know where that is, right? And there is like thousands of ways to Rome, and that might be one of them to maybe hopefully. Oh yeah, sure does. Yeah, no, that's true. Good point. And a Mexican beer. Is there any Italians in the room here? Okay.
So when you're doing your single cell sequencing, and you had that picture of the large bulk of the tumor. Are you trying, I don't know, of course there's fiscal and feasibility issues here, but are you doing multiple samples out of that tumor bulk to account for, because it would be very interesting to compare, because you talked about heterogeneity at the beginning, to compare those fields of sample tissue?
Right. No, not yet. So we have about $500,000, which is enough for 30 patients. No, Tim and I had talked about it. Maybe we could do it for one patient, but if you start talking about 3,000 RNA-seq, no, we would love to, but right now we're not. But no, definitely that would be one thing that, I think it would be helpful to do. The one other thing I haven't told you, and that's still all work in progress. And if you guys have any suggestions, ideas, I mean, we like I said, we only have done 2 patients now. The one thing that we just recently changed, so with the first patient we flow sorted essentially just all the immune cells out, because we we said, well, we can have 96 cells. We want to make sure we get at least stroma cells and tumor cells. So we just changed that so that we're actually doing 2 patients today and then 1 tomorrow. And we're not going to flow sort them anymore because now we can do 3,000 plus cells. So we actually are quite interested even in the immune cells. There's some work I've seen recently in breast cancer where they found in the infiltrating immune cells JAK2 mutations, I think, in those T cells that, I mean, why wouldn't that be in ovarian cancer? So that, I think, in itself would be very interesting to look at. So hopefully we can actually capture some of those.
The other thing might be, again, it's a feasibility and money issue, but cells that associate with the omentum versus cells that are associating with the mesothelium, the peritoneal cavity, you know, it's like you may start to get some really good ideas on that metastatic.
And we're trying to, we have a couple we're trying to do. Sometimes it comes down to what's the most viable tumor that we can get because we have to keep those cells alive. But yeah, we're trying, I mean, we'll record all that, whether that was from primary tumor versus the last patient, for example, we've taken the omentum with that idea. We had some issues, take rates with the avatars. The omentum, so those cells that are in there, they're really using the fat tissue as essentially the energy source. And if you put those cells sub-Q, and those they might survive better, and they already have proven they can survive outside that primary tumor. They already have that sort of that check mark too.
Okay, so you're really, really concentrating on sort of the largest mass of...
Yeah, the most viable right now, but we will have differences. So there will be primary tumor, there will be some from the omentum that we can then sort of like retrospectively, hopefully, sort of sort out differences. But yeah, you know, you're absolutely right. I mean, in an ideal world, we can do the primary tumor and 3 other ones. Anybody has $1.5 million or so? I mean, I'm happy to.
So, the concept of your the tumor subtyping, right? And that's all done at diagnosis, right? And then you showed us that all subtypes potentially exist right there.
Right.
Is there?
And we knew that from if you go. I didn't show you that, but so. When those subtypes get assigned, and again, I'm not a bioinformatician, so is anybody in the room, you go ahead and correct me, but essentially you assign a probability to one subtype. It's not absolute. So they all have different subtypes. We knew that before, but we've never seen that clear, that really there's this group that's probably that part and that, which could already be because some of those are stroma cells and some are cancer cells. That in itself will probably already give you sort of different subtypes.
I guess my question is, do you know whether when patients recur, they retain the same subtype? Is there any information on that?
It seems to be most of them, but there isn't a lot of work done on that. So that's one of the other things we're trying to look at because we actually, there's one study I mentioned, the GOG213, there's hopefully validations that we can get. That's in the recurrent setting. So we might be able to get some actually primary tumor and then, yeah, that's a good question. But some work that has been done through what Paula Luska at Mayo originally did, and now John Baroa has taken over, their avatar models seem to suggest that they retain their subtype. But it's possible if they have different subtypes and that you select out, let's say, the worst, like the mesenchymal. So yeah, certainly possible. But so far we think they retain that, at least some of them.
And I think also just to what you just said about the omental or source versus the primary tumor source, there is... I don't know, admittedly anything in ovarian cancer, but I know in leukemia there is some data coming out that the fat niche is actually providing those survival signals that allow the stem cell to survive chemotherapy. So,
yeah, there is some Ernst Lengel's group out in Chicago, so they've actually shown that. Yeah, no, there's definitely. So one thing is fat has the highest amount of energy per gram, so that makes sense that you evolutionary want to take what has the most energy in the belly. But yeah, no, there is more to the omentum for sure that does support the tumor.
Yeah, and there are actually, some of those signaling pathways, not just energy source, but actual signaling pathways have been worked out, at least in leukemia, or at least started to.
That'd be actually interesting. Yeah, we should definitely look at that and compare.